Oh, hello, everybody. We are here with Dr. Gemma Gomez-Giro. So I have the pleasure to present her because we did together a PhD on Batten disease in Luxembourg. So this is the nice thing to work in a rare disorder that at the end you make friends over the years because the field is so small. So Gemma, please, can you tell me how was your experience working on CNL3 and to develop a PhD project on this topic?
Um, yes, of course. Hello, everyone. I'm happy to be here sharing my experience in building a PhD on Batten disease. And well, my project on Batten disease had two main goals. The first one was to create cell lines to study the disease. And what do I mean by that? And why was this necessary? So creating cell lines was necessary because Batten disease is a rare neurodegenerative disorder that affects children mostly. And to be able to study the disease, we need their cells. And sometimes this can be problematic to get them. So an alternative to this would be to get cells healthy from a healthy adult, for example, and introduce the genetic changes that cause this disease in children. And this was the idea to be able to give us tools to study the disease. And then the second goal was to study the disease and how it affects brain development in these kids. And for that, we used organoids as a technology. And organoids are basically three-dimensional structures made out of these cells that we created, and in our case end up resembling the human brain. And like that, we could study the early changes that would happen in the brain of Batten disease patients.
And
And
what actually you see in your model? So did you were able to see this lysosomal storage phenotype that has been described in the patient cells?
correct, yes. So that was the nice thing about it, that in our small model of the human brain, we were able to see these classical deposits that are present in the brain of these children. So this lipofuscin accumulation and so on, and
Mhm.
some other changes. So
Quite cool. And so now you are working on more in Parkinson's disease, so more common disorders. Did you see any difference to study rare disorder comparing to more common disorders?
well, yes. So already the fact that there are less people working on rare disorders and more on a field like Parkinson's disease is a big difference. And yeah, it's a much bigger community that studies Parkinson's disease, and there is a lot more explored, and there is a lot more unexplored on rare disorders like Batten disease, and that can be sometimes a challenge. Of course, we tried to build as many collaborations as well as possible, but as you mentioned before, it's a small community, so the rare disorders field. So in the end, it's a bit of a family, no? Whereas in Parkinson's disease, the feeling is a bit more. Everybody does their own thing and hopes for some that at one point everything converges together. Whereas I have a feeling in the rare disorders field, there is a bit more this community feeling of achieving something also together, no? And of course, the big difference is that there are more resources allocated to Parkinson's, to diseases like Parkinson's or Alzheimer's that affect
Mhm.
many more people.
So, why you started actually your PhD in Batten disease? How you come to this idea? So, what was your main motivation, or you decide why decide to do this PhD?
Um, well, I wanted to do a PhD on neurodegenerative disorders. That was one thing. The idea of studying an actual rare disease motivated me, especially because I felt that the impact maybe that my research could have if I managed something um, would be greater in a way. So my motivation was really that I wanted to help these affected kids. In the end, the my PhD was more dedicated to help actually other researchers maybe to study the disease by generating these lines and by exploring them with the organoid model and so on. But in the end, I think the ultimate goal is always to help the people, the children in this case.
And one last question: which was the main? So, which role played the patient association in your PhD?
The role of patient associations was vital because they were actually mostly funding my studies. So I actually I have to thank them for that, of course, and I was very I was very happy to actually meet them in meetings and conferences, like the involvement of the parents that really are looking for uh, better treatment or, of course, hope for a cure for this disease was actually very important, and they were very present, actually.
Okay, thank you very much, Gemma, for sharing your experience. And unfortunately, you leave us. You are not working anymore in our field. But let's hope that you come back to the Batten disease field. You never know.
One never knows, yes. I would be happy to.
Thank you for accepting this. So have
Thank you for
a nice
having
day.
me. You too. Thank you for having me.